Let's review the inheritance patterns that we've been studying for the last few weeks so that tomorrow in class we can solve problems that address many of these different patterns with relative success. So, as you're watching this slideshow, you might want to take notes in this format. Here's a sample table that could help you keep track of the differences between the patterns that we've studied. You don't have to use this table, but I urge you to take notes in some fashion. All right, so let's review. Which patterns have we studied? First of all, there was complete dominance on the autosomes. This involved two different alleles that could produce two different phenotypes, and we looked at this in one trait crosses, two trait crosses, and multiple trait crosses. We also looked at complete dominance on the sex chromosomes, sex linkage. This had two different alleles and two different phenotypes. Then we looked at incomplete dominance. Again, two different alleles, but three different phenotypes resulting. And finally, we looked at codominance, where there were more than two alleles and multiple phenotypes possible. So let's start with autosomal complete dominance. So here we're dealing with chromosomes 1 through 22, and simple alleles that are either completely dominant or completely recessive. One of the crosses we looked at was purple flowers versus white flowers. We've got one trait, the trait of flower color, and if we cross these, there's two possible phenotypes. You can either get purple phenotype or you can get a white phenotype. There's three possible genotypes. You could have homozygous dominant, also known as true breeding. You could have heterozygous dominant, also known as hybrid, and you could have homozygous recessive, also known as true breeding. We also looked at two trait crosses, in this case the P's, which had the trait of color, yellow versus green, and the trait of shape, smooth versus wrinkled. And solving these traits worked the same as one trait crosses, you just had more gametes to keep track of. Now there were some shortcuts and important ratios to keep in mind, and in order to keep those in mind, we have to make sure we know the difference between monohybrids and dihybrids. All of these genotypes listed here are monohybrids because there's one trait, we can tell that because there's one letter, and the parent is heterozygous for that trait. Dominant, recessive, dominant, recessive, dominant, recessive, dominant, recessive. Dihybrids are heterozygous for two traits. We can tell there's two traits because there's two different letters. So heterozygous and heterozygous, heterozygous, heterozygous. You get the picture. So let's take a look at some of the ratios we get for a monohybrid cross. Here we've crossed two purple flowers. They're dominant, but they're heterozygous dominant. They've each got one dominant, one recessive allele. When we cross them, we get an interesting pattern every time. We'll see that for the genotypes, there's always one homozygous dominant, two dominant P's, two heterozygous dominant, one recessive, and then one homozygous recessive. And you can see that in the Punnett square. 1, 2, and then 1. We also get an interesting phenotype ratio. For our phenotype ratio, we see that 3 of the 4 have the dominant trait, which is purple, and 1 of the 4 has the recessive trait, which is white. So make sure you keep straight genotype, which is the actual alleles that the organism has, versus phenotype, which is the trait that the organism expresses. Here's another way of looking at that monohybrid cross. Again, for phenotype, 3 purple to 1 white. For genotype, 1 to 2 to 1. What about dihybrids? Well, dihybrids result in a very famous phenotype ratio. If you cross two parents who are both heterozygous for both traits, and you can see that here and here, you'll get approximately 9 with both dominant traits, 3 with one dominant, one recessive trait, 3 with one recessive and one dominant trait, and then this last one actually is a mistake. I won't tell you what it is, but it should actually be a different number there. Should be a 9-3-3-1 ratio. All right, one more thing about simple dominant traits. If we've got a multiple trait problem like this to solve, it's a big headache to make a huge Punnett square, so remember the rule of multiplication. So let's say we're crossing two organisms that are trihybrids, and we want to know what's the chance of dry earwax, which is recessive, dimples, which is recessive, and 5 fingers in the kids. Well, we can just solve one trait at a time. The chance of dry earwax on its own would be 1/4. The chance of dimples on its own would be 1/4. And the chance of 5 fingers is 1/4. So to get the final answer, the chance of all 3 of them happening together, we simply multiply. And without making a giant Punnett square, we get that there's a 1 in 64 chance of all 3 recessive traits. All right, let's move on to sex linkage. Sex-linked dominance explains why men have certain traits more often than women, traits like colorblindness, hemophilia, and baldness. And this has to do with the fact that the genes for these traits are on the sex chromosomes, and men are missing a portion of the Y chromosome. So let's take a look at an example of a problem here, colorblindness. If we've got a mother that's colorblind and a father who's normal, what's the chance that the child will be colorblind? The trick to solving these crosses is to remember that you have to show the X and Y chromosomes. If you forget to do this on a problem, you're going to get the wrong answer. So when we set this up, here's our mom's genotype. She's recessive. Here's the dad's genotype. He's dominant, but keep in mind, because his Y is short, he doesn't get an allele here. And when we set up the Punnett square, there's the mom, there's the dad, and here are the offspring. And please note that the men are expressing the colorblindness trait while the female offspring are normal. And this is very common with sex linkage. It's a big clue. If the males express a different trait than the females, it should trigger an alarm that you're dealing with sex linkage. All right, let's keep moving. Incomplete dominance. Incomplete dominance can look like complete dominance, except when you get an interesting third phenotype. That's usually the intermediate. Here we've got a red and a white flower crossed. When we cross them, we get pink offspring. So that's our clue that we're dealing with incomplete dominance, because that dominant R does not totally overpower the recessive R. Instead, we get a mix or a blend in the phenotype. If we were then to take this heterozygous pink flower and cross it with another heterozygous pink flower, we would end up with these results. And actually, these numbers should look familiar to you. We've got, again, the 1:2:1 genotype ratio, but we also have a 1:2:1 phenotype ratio. So that's how you can distinguish this from complete dominance. If you remember, with complete dominance, we had a 3:1 ratio. But here, because of that intermediate phenotype, we get a 1 red flower, 2 pink flower, and 1 white flower ratio. All right, finally, codominance. Codominance, our usual example is blood groups. And with blood groups, we have 2 different dominant alleles, A and B, and then our recessive allele, which was the little I. And if we put these together, we can get a range of genotypes and phenotypes. These possible genotypes will give you this phenotype on the red blood cell. And we call that phenotype blood group A. If we have a combination of the dominant B alleles or the dominant B and the recessive allele, we get blood group B. Here's where things are really interesting, because now both A and B can be expressed at the same time. So it's codominance as opposed to incomplete dominance, and we get AB blood group. Finally, 2 recessive alleles give us no carbohydrates on the surface of the red blood cell, and this phenotype is blood group O. And that concludes our review of all the inheritance patterns. Please make sure your notes are complete so that you can come into class ready to solve a variety of problems.